All right, let's dive right into this explainer and declassify the files on what is arguably your body's most ancient, ruthless, and most misunderstood defense system. I mean, we constantly hear about antibodies and white blood cells, right? But today, we're focusing on the covert operations happening in your bloodstream literally right this second. So here is our briefing. We'll start with how this defender got misnamed, then meet C3, the ancient weapon. We'll look at its 3 paths to activation, the incredible membrane attack complex, how we prevent friendly fire, and finally, what happens when the shield fails.
Okay, Section 1, the misnamed immune defender. Let's uncover the origins of this biological strike force.
You know, when this system was first discovered way back in the late 19th century, scientists actually gave it a pretty epic name. They called it Alexine, coming from the Greek word Alexis, which translates to defender or to ward off. I absolutely love that, because it perfectly establishes the high stakes of what this system actually does. You know, right from the start, it was recognized as a primary protector.
But then things shifted. The early view basically framed it as a sidekick. They assumed it merely complemented the overall immune response, kinda acting as a simple bridge to help antibodies and neutrophils engulf pathogens. And while that's actually why they ended up renaming it complement, but the modern reality is so much cooler. Today, we know it's this massive, totally independent network of around 30 proteins, acting in a super rapid domino-like cascade. One protein activates, which turns on the next, and then the next. It doesn't even always need antibodies to start the fight. Honestly, they probably should have stuck with Alexine.
Moving on to Section 2, let's meet C3, the ancient weapon and original antimicrobial force. So C3 is definitely not just another cog in the machine. This specific protein has been around for hundreds of millions of years. It's the original antimicrobial force, and it's present in almost every single invertebrate on Earth. But what makes C3 so incredibly special is that it contains this hidden magic bullet tucked inside its folded protein structure. It's called a thioester bond. This specific combination, a sulfur atom and a carboxyl group, is so powerful that it can just rip apart and disrupt almost any fatty acid. And yes, that includes the membranes of invading microbes. It is literally a loaded weapon, just kept safely folded up until it's time to strike. Now, the crucial point here is exactly how this weapon deploys when activated. When triggered, it splits into a 2-part special ops mission. Part 1 is the larger piece, C3b. This acts as both the destroyer and the docking station. It forcibly attaches right onto the invading microbe, unleashes that destructive thioester bond onto the membrane, and at the same time, it grabs hold of a passing neutrophil to totally set the microbe up for destruction. But then there's part 2, the smaller piece that breaks off, C3a, doesn't just fade away. Nope. It drifts off to act like a biological flare gun. When it hits immune sentinels like mast cells, macrophages, or even the liver, it sounds a massive alarm, causing your body to just flood the area with more defensive proteins. Talk about teamwork, right? All right, Section 3, the 3 paths to activation, or how exactly this sleeper cell is triggered. What's truly remarkable here is that there are 3 totally different biological roads, but they all lead to the exact same explosive activation of C3. So first up, we've got the classical pathway. This was actually discovered first. Basically, an antibody spots an invader, binds to it, and calls in this specialized strike team known as the C1 complex. But hey, what if there's no antibody around yet? Well, that is where the lectin pathway steps in. See, a lot of microbes have this specific carbohydrate on their surface called mannose. When our immune system bumps into that mannose, it directly triggers complement components C2 and C4, totally bypassing the need for an antibody. And finally, my personal favorite, the alternative pathway. This is actually the most ancient method. So get this, our C3 protein will occasionally just spontaneously open up and grab a water molecule right out of the blood. Normally, it just self-destructs in milliseconds, but if there happens to be an invading microbe nearby, and some liver proteins, specifically factors B, D, and properdin, are floating around, well, they bond together, stabilize that watery C3, and violently initiate the cascade. It's like a trap just waiting to be sprung.
This brings us to Section 4, the membrane attack complex, the ultimate biological weapon.
So this incredible microscopic machine literally assembles itself right on the surface of a pathogen. Following the activation of C3 we just talked about, another protein, C5, gets triggered and splits in half. Its larger half, C5b, becomes the foundation. It quickly gathers up other complement members, specifically C6, C7, and C8, building this towering structure on the enemy cell. And then, the finishing blow.
Component C9 drops into place, oriented perfectly at a 90-degree angle, swinging down like a hammer to push the entire massive aggregate deep into the pathogen's membrane. It literally punches a physical hole right through the cell's armor. No way for the pathogen to hide from that. And the result of that hammer strike? Utterly devastating. The membrane attack complex, or MAC, creates this wide open channel. Suddenly, there's a rapid, totally uncontrollable two-way flow of water and solutes. The microbe experiences a massive imbalance in its pH and nutrient levels, which just completely destroys the integrity of the cell. Death to the microbe is basically instantaneous. Now, here's a really fascinating detail. This system destroys bacteria, fungi, and parasites beautifully. But viruses actually escape this specific complement attack. Why is that? Well, because viruses don't have traditional cellular membranes. There's nothing for the C9 hammer to actually punch through. Which naturally leads us to Section 5, preventing friendly fire, or how our own bodies actually survive this crazy cascade. I mean, think about it. If this incredibly destructive MAC system is constantly floating around in our bloodstream, and it can activate spontaneously with just a random water molecule, why doesn't it kill us? Well, first off, we have to understand just how targeted this system usually is, thanks to a process called opsonization. Derived from the Greek word for to prepare for eating, opsonization is basically how the complement system paints a giant target on a pathogen. You see, all cells, both ours and the invader's, naturally have a net negative electrical charge, so they repel each other, kind of like two identical magnets. Without this complement bridge acting as an opsonin to firmly link the immune cell to the pathogen, our white blood cells would waste a massive amount of energy just trying to chase down and grab onto cells that are actively repelling them. So the system is beautifully designed to highly target its prey, focusing all that destructive energy far away from our own healthy cells. Furthermore, our own host cells are fully equipped with specialized biological shields. These proteins actively dismantle the complement weapons if they ever accidentally land on friendly tissue. Take CD59, for example, which is perfectly nicknamed protectin. If a membrane attack complex accidentally starts building on one of our own cells, CD59 just swoops in and essentially removes that C9 hammer. It totally disarms the weapon before it can punch a hole. We also have factor H. This protein constantly patrols our serum and cell surfaces, guarding specifically against that spontaneous alternative pathway we discussed earlier. So if C3 reacts with water, but there's no microbe around to fight, factor H just neutralizes it immediately to protect our tissue. It's a pretty flawless failsafe. But what happens when that failsafe breaks? Let's look at Section 6, when the shield fails, and the real-world stakes of genetic defects. To really grasp the stakes here, take a look at this specific pathogen, Neisseria meningitidis. It is a nasty, nasty bacterium that tries to invade by colonizing the surface lining of our respiratory tract, and then it tries pushing straight through to the tissues underneath. Normally, a healthy innate immune system, specifically our complement cascade and neutrophils working together, will arrest this invader right there on the spot. The MAC punches holes in it, the C3b tags it for eating, and honestly, it's game over for the bacteria before it ever even reaches our bloodstream. But a tiny genetic defect can change everything. Between 1 in 20,000 to 40,000 people. That's the estimated number of people who have a genetic defect in components C5 through C9. And because they lack the ability to fully form that membrane attack complex, they simply can't drop the C9 hammer. This leaves these individuals tragically vulnerable to this specific pathogen. It allows the bacteria to spread into the bloodstream and settle right into the meninges, which can lead to fatal bacterial meningitis. To put it in perspective, this single statistic brilliantly illustrates just how heavily we rely on this microscopic defense network every single day. It truly is an incredible, highly coordinated, ancient killing machine that keeps us alive without us even realizing it's there. From spontaneously grabbing water molecules out of the blood to dropping microscopic hammers on bacterial membranes, your complement system is always on guard. So, the very next time you manage to fight off a sudden infection, leave a little room to ask yourself, just how well is your Alexian working today?